Welcome to today's webinar, Trauma and the Brain, the Biological Basis of Enduring Effects and Interventions with Anthony Pravatsky. I'm going to do a couple of housekeeping announcements here, and then I'll introduce our speaker. If you'd like to ask a question during the presentation, just please use the question feature, which is on the right side of your screen, and we will try to do our best to answer those at the end of today's webinar. If you're interested in earning 1 CU, just be sure to stick around to the very end. We'll have a code that's given, and you'll need to email me that, and we'll have more instructions at the very end of this presentation today. We are recording this webinar, and we will upload the presentation to our website. And lastly, if you are on Facebook, we frequently post, webinars and upcoming trainings on our Facebook page, so just be sure to check us out there if this is something that is of interest to you. So, Anthony Pravatsky received his bachelor's degree in neuroscience from Trinity University, and he's currently pursuing his medical and PhD degree at UAMS. He's spending a lot of time in the UAMS Department of Psychiatry's Brain Imaging Research Center, and his current research interests include fMRI analysis and exposure-based therapies for PTSD. And I'm going to turn the webinar over to Anthony and let him introduce himself and his topic a little bit.
All right. So thank you. Thanks, Chad, for the invite to do the webinar. It's a real honor. I haven't done a webinar before, but I'm really looking forward to it. I'd just like to note that I have a past of working in some molecular research as well, so you might notice me going a little molecular in this talk. I hope I hope I can condense it down and not make it too overly complicated. But if anyone has if you think that I glossed over something or if something's not clear, I'm very happy to answer questions. So just be aware of that. Again, thanks for the invite, and let's see. So I have control now. Another thing I'd like to add before I start is, like Chad said, I'm doing research right now on a potential new enhancer for exposure therapy in PTSD. It's a very cool study. I can provide more details if you're interested at a later time. I know most of you work with adolescents or children, but this is a study in adult women with PTSD. We'll actually screen them to see if they meet some initial criteria before they come in for more screening. But if you're interested, let me know. We really need more participants. We've had about 30 adult women participate so far, and we're really looking forward to getting some good data out of that. But I'll return to that at the end as well. So I wanted to start today with, although you all are exposed to this every day, but I just wanted to review some of the statistics about the costs of trauma. And this will kind of set the stage for understanding why it's so important to understand the neurobiology of trauma. And also part of setting the stage for that is understanding the acute stress response and understanding how the acute stress response differs from the long-term effects of stress. And that also will kind of help with understanding how certain interventions work to interact with the stress process in an adaptive way. So I'll go over the acute and the lasting effects. I also hope that by the end of the talk you'll at least be more familiar with certain laboratory models of studying the effects of trauma or stress. And also, some of this will be a review, I'm sure, but I'd like to cover some of the factors that moderate the effects of stress. that can either make someone more susceptible or more resilient. And those could include things like development. We'll go over some of the biology of brain development. Those could be genetics. Those could be past experience, so environmental factors. And this is by no means going to be a complete talk about the biology of trauma. But I covered some of the points that I think are personally some of the most interesting or compelling points. And then I'd like to go into the neurobiology of treatment and how certain treatments that are known to be effective, just sort of the biological basis for those effects. That's kind of the platform by which we try to improve, or at least in a disciplined way. Knowing the biology of the treatments is a very good basis for actually investigating new forms of treatment. So first, like I said, I'd like to cover just some of the personal effects of trauma. What are the costs? Many of you may be familiar with this study. I would hope that you are. So I apologize if it's too much of a review, but this is a very interesting study that they did out in California in 2006, where they found actually in I believe it was an HMO, an established medical organization with a large database of patients, and they retrospectively looked at 17,000 adults, and they studied just some categorical factors that may or may not have happened in their early life. And they wanted to see, this was, what sort of compelled this was Vince Felitti, who was a physician in this system, with actually, he said, no interest in research at the time, and really particularly not really interested in mental health research. But he was, I believe, an internal medicine or primary care physician, and he was just started noticing that he had many of his patients who were severely obese or were smoking. He seemed that he thought it was just uncanny how often these patients reported some sort of abuse during childhood. And that got him very curious, despite his lack of research work. And so he decided to look at this database, and it may not have been retrospective, but anyway, he had them answer just in these categorical questions, whether they experienced emotional abuse, physical, sexual abuse, whether they had someone using alcohol or other drugs in the house growing up, whether they're exposed to someone with a mental illness when they were growing up, whether they had their mother or mother figure caregiver was exposed to violence or abuse, whether there was criminal behavior in the household, and whether there was some sort of parental separation or divorce. And these were scored very simply, just yes or no. Did you experience this? And the sum of that was used to predict 18 outcome measures of interest. So depression, anxiety, hallucinations, any sleep disturbance like insomnia, severe obesity, somatic symptoms or somatoform disorders, current smoking, alcoholism, any street drug use, any injected street drug use, so that's just designed to be another categorically greater drug abuse type, whether they have report impaired memory of certain aspects of their childhood, whether they had early intercourse, whether they had promiscuity. I believe that was measured as 30 sexual partners or more. whether they reported sexual dissatisfaction or high perceived stress just in general, whether they reported difficulty controlling anger, and also if they seem to display a high risk of perpetuating interpartner violence. And it's just super compelling. You do not see relationships this strong in many areas of science, but, or at least in the mental health field, but there was a direct linear relationship between the number of categories of trauma that you had in childhood and the number of these outcome comorbidities that were reported. And so one thing you might ask is this just causation? Is this just correlation, or is that actually a result of trauma? I'll get into that more throughout the talk, but something good to keep in mind, of course, because it's a very important factor. But I just want to look at mental health disturbances at this point. So just as an example here, if someone, so you have a baseline, about half of these patients who didn't report any sort of adverse childhood event in these categories. For example, if they had just the standard rate of panic reactions is 8%. If someone had an A score of just one of these categories, it moves up to about 11%. And by the time you report 4 or more of these adverse experiences, you are at a rate of 21% of those individuals report panic, or they're 2.5 more times more likely to report that. That's what this 2.5 odd ratio is. For depression, someone with 4 or more of these adverse experiences is 3.5 times more likely to report depression. So up to half of those individuals report it. Whereas in the normal population, it's more like a fifth. Anxiety, you're 2.4 times more likely to experience anxiety with 4 or more of those, and 2.7 times with hallucinations. I thought that one is compelling because we don't typically think of something like hallucinations as a categorical outcome of childhood trauma, but clearly 2.7 times more likely. If you've had 4 or more of these experiences, that's a pretty big deal. Now, also here, like I originally said, Vince Folletti, who was one of the original creators of this study, was interested in looking at obesity, and someone is 2 times approximately more likely to have severe obesity if they've had 4 or more of these childhood experiences. 2 times more likely to have some sort of sleep disturbance. And also something I'm sure we all hear a lot about, very much more likely to use illicit drugs, 4.5 times more likely to use illicit drugs if you've had 4 of these experiences. Even one of these experiences makes a person, in this data at least, 2 times more likely to use an injected drug. And that moves up to 11 times more likely to, Vince Folletti is fond of labeling this as self-medication. I think that's totally correct. But let's move on here. What is the biology of that? I mean, is the trauma actually causing these effects, or do people growing up in those hard situations just come from a line of having those problems anyway? So let's just first review the acute stress response. Trauma can be viewed as just causing an extreme stress response. So what that is, it could be called your fight, flight, or freeze reaction. So this is in contrast to the rest and digest reaction that you all might have heard of. So the fight, flight, or freeze reaction is caused by sympathetic nervous system arousal, whereas the rest and digest autonomic response is part of the parasympathetic nervous system response. Is there anything really wrong with this? No, because evolutionarily, this is built into us to prepare us for adaptive behavior. If we see an animal that's a threat, or if we have some other threat in the environment, we need we need some adrenaline, so norepinephrine. We need some cortisol to kind of, that recruits our blood sugar to get our energy levels up. And this is all really done through, well, there's a lot more to it, but the basics of this is that it starts in the brainstem, and it moves up to the midbrain, where the hypothalamus releases corticotropin-releasing hormone, CRH, and that acts upon the pituitary, which then releases adrenocorticotropin hormone, and that then acts on the adrenal glands to release cortisol. But like I said, there's a lot more to this, and that's really important for understanding the effects of trauma and why susceptibility can be such a variable outcome. So if you just take a broad look at what the brain structures are involved in the stress response, there are 4 primary aversive stimulation inputs, if you will, to the stress response. So part of those come through the habenula, which, not to get too technical here, but the habenula is involved in sensing kind of negative outcomes or expecting negative outcomes. And the LC, the locus coeruleus, is the main brain center for synthesizing and releasing norepinephrine. And then the stria terminalis is a primary communication route to the amygdala. But interestingly, one of the primary effects of the stress response is that it activates the dorsal raphe, or the DRN nucleus. Now, that might be kind of surprising, because in popular culture, people kind of like to consider serotonin to be the feel-good relaxation hormone, but things are a lot more complicated than that. So what happens, there's a lot of biological research to back this up, is that anytime there's a stressor in the environment, like a fearful stimulus, you have this activation of serotonergic neurons, and specifically the dorsal raphe neurons, which are a region in the midbrain. They release serotonin to many other structures in the brain. And so acute stress causes an acute increase in serotonin to many different structures. And that leads to many of the negative outcomes that we see. So the amygdala, which in popular terminology, if you will, we kind of consider the amygdala to be the center for fear. But again, with many things in the brain, it's more complicated than that. But bottom line is that this activation of this key midbrain structure and its subsequent innervation of other brain structures, it occurs with the acute stress response. And what I want to stress is that this is a very automatic process. So it doesn't really leave us much room to consider how someone might respond adaptively to a stress response. But one other thing I want to highlight, and the importance of this diagram will come up later in the presentation, but I would like you to have just a basic, at least, understanding of the importance of it. One other thing I wanted to highlight is the stress activation of this midbrain structure, the DRN, also directly influences the nucleus accumbens, which is a part of the brain involved in processing reward, and it's kind of a key integral part of the brain that integrates many other signals to actually create approach and avoidance behavior. So just keep this in mind, that the stress response directly affects this reward-related structure. And as I said, the DRN, the dorsal raphe nucleus or nuclei, they innervate many other structures. And what I want to highlight with this diagram is that some of those important structures are, again, the amygdala, the hippocampus, which I didn't focus on explicitly in this presentation, but it will come up, or I will bring it up again and again as a very important factor. And then, of course, you see just the entire rest of the brain is just bathed in serotonin from these regions. But another very important region not to forget about is the DRN actually has direct projections to the hypothalamus as well. And the hypothalamus has, so it has serotonergic receptors, and that will become important later as well. And then the DRN also innervates the pituitary. And as I said before, the hypothalamus and pituitary are key to this hormonal reaction to stress. Now, so that's the basic biology, but how can we actually measure how this process works in the human brain? A lot of this research came from rodent studies where you can actually do some things that you can't do to humans, obviously. But one of the most interesting and earliest investigations of this process in the human brain is actually an fMRI. So this paradigm is, it's used in many different studies, but the important concept behind this paradigm is that we're trying to understand how the brain processes threat responses and then sort of learns how to deal with a threat or something that might be perceived as a threat but is actually not harmful. So participants might be presented with a series of faces. Intermittently, they might have a fearful face in the context of a bunch of different happy faces. And a very common behavioral finding is just that the brain, it pauses when confronted with a fearful stimulus. And so just on average here, the reaction time to participants in this paradigm are told to make a selection if they see a fearful face, but not if they see a neutral face. And so they're kind of having to do this goal-directed activity in the context of knowing that there might be this sort of fearful stimulus. And so their average reaction time to actually approach or commit this goal-directed behavior when they see the fearful face is on average longer, which implies that the stress response system is somehow interacting with and affecting the goal-directed brain system. Now, what is the actual neural basis of this? Well, you can do a simple test to actually show that this greater reaction time toward fearful stimuli as opposed to happy stimuli. So you can also have them, say you have a happy face in the midst of a bunch of neutral stimuli. Well, what on average people do is they respond more quickly to a happy face. But anyway, so the interesting factor here is the time it takes to respond to fear stimuli or engage in goal-directed behavior in the midst of fear over and above engaging in goal-directed behavior in the midst of no fear. So what we find is that this increase in reaction time is directly predicted by how activated their amygdalae are. And, oops. And contrarily, the opposite, so a brain structure that predicts a faster reaction time is the ventromedial prefrontal cortex, which is it's a regulatory brain region, which it has direct inhibitory connections to the amygdala. So this is just a basic example showing what the biology is of that automated response in humans and the inhibition of that automated response. So theoretically, throughout this task, someone should become habituated to this fear stimulus and actually have faster reactions each time, because they sort of learn that, oh, this is not something to get hung up on. It's not actually threatening. And so what the brain region is that this data suggests is actually regulating that adaptive response, and the better goal-directed behavior is the excuse me, ventromedial prefrontal cortex. And then going along with this, like I said, there are we know from animal research that there are direct inhibitory projection neurons going from the prefrontal cortex to the amygdala. And so what this is just reiterating in humans is that that decrease in amygdala habituation is actually predicted by how strong the co-activation of the prefrontal cortex and amygdala is. In fMRI, we call functional, well, basically what functional connectivity is co-activation of structures. So in this case, that co-activation is an adaptive thing. So now that I outlined just the basics of the acute stress response, I want to go into how does this even relate to the enduring effects of early life stress. Well, we can look at some very obvious factors like hormones circulating in the body, of course. We can also look at brain structures. So some very common findings are that people with early life stress have lower cortisol at baseline and higher norepinephrine. That's a little counterintuitive, but I'll get into why that's important on the next slides. Also, something commonly found in people with trauma is reduced hippocampal volume and reduced amygdala volume. The hippocampus, I'll touch upon this later as well. It's one of its important roles is thought to be that it regulates it regulates regulatory responses by sort of giving a proper context to what is going on in the environment. So as you can imagine, if your hippocampus is not functioning as it should, then you'll kind of have this failure to properly regulate, whether it's your fear response or whether it's inhibiting that fear response. The hippocampus has a role in all of that. And one thing to note, I don't have it explicitly in the presentation, but the hippocampus is directly negatively affected I believe, by just purely biological processes where negatively affected by the stress response, where cortisol itself, so the stress hormone released during acute stress, high doses of that can actually cause neurotoxicity and reduce hippocampal function. So that may be why the hippocampus is observed to be smaller in early life stress. However, we also have evidence from animal experiments that that is directly true. And something that kind of goes along with that is that another thing found in this case, women who are sexually abused as children, is a deficit in verbal declarative memory, and that's most likely related to hippocampal dysfunction or atrophy. And that also reflects that outcome measure in the adverse childhood events study, where those with greater reported amounts of trauma reported also greater gaps in memory of their childhood. And another important long-term effect of chronic stress is that it actually causes a direct sensitization of the dopamine system. So interestingly, stress itself causes a surge in dopamine in key regions of the brain, and repeated instances of that are, strangely enough, they affect the brain in a way that's similar to repeated instances of drug use, which typically addictive drugs also release dopamine. So you have this negative feedback where stress itself can cause surges in dopamine and sensitization of that system, making potentially more impulsivity and more drug-related craving, which can lead to further drug use, which can sensitize the system to stress, which then sensitizes to further drug use. So it's just a terrible cycle of positive feedback. So we see these obvious effects in biology, our structural and hormonal systems. But what we also see is that early life stress even changes just instances of behavior. So not even just like long-term behaviors like drug use or impulsive sensation seeking or depression or anxiety, but even in just short-term paradigms, early life stress can affect behavior. So, it can have enduring effects on behavior. So in this case on the left, this is actually from an animal model of early life stress where they tried to control for these factors and actually show that early life stress has causative influence on these negative outcomes. So what they did is they had one group of rodents that was just reared normally, and then one group of rodents that was exposed to early life stress. And what they found is that contrary to healthy adolescent, or healthy pre-adolescent rodents, whatever you want to call that, I guess they're a couple weeks old. So typically, healthy rodents will have this, don't worry about understanding exactly what this is, but it's a measure of neural activity in the amygdala, a molecular measure. Typically, the amygdala response is great in sort of adolescent congruent rodents in a stressful paradigm. But the pre-adolescent and adult rodents have this lower amount. But in animals who are chronically stressed very early in their life, later in pre-adolescence at test, in adolescence at test, and as adults at test, they display this consistently high level of activity in the amygdala. And despite the fact that on the right-hand side here, this just shows that as a group, regardless of whether the animal had early life stress or not, activity in the infralimbic cortex, which is the analog of the prefrontal cortex in the rodent, activity in that region increases with age, even in the early life stress rodents. However, it's as if they're not able to dampen down this, this stress response indicated by amygdala activity. And we also can see just in instantaneous experiments in humans with early life stress of some sort. I believe in this case the early life stress was the unfortunate circumstance of individuals who were in an orphanage in some third world country where they had very, very poor care and not a lot of direct human interaction. And they had these individuals who are now adults, and I don't recall if it was in the United States or not, but anyway, they had these individuals with this terrible early life stress, and what they found is that in a paradigm similar to the one I talked about earlier, those individuals who had this early life stress displayed a much higher reaction time. So they were failing to inhibit a stress response, an acute stress response, in order to engage this goal-directed behavior, which the measure of reaction time. And then you also see this reflected in their brain. So if you scan them in an fMRI paradigm, you actually find that the individuals who had early life stress, despite that they're being exposed to the exact same stressors in the fMRI environment, they have this long-lasting, and by long-lasting I mean seemingly for life, reaction where they have a greater stress response, just like we saw in the animal models. But so I painted a pretty bleak picture, but there are things that moderate these effects, fortunately. So one interesting factor is the very important distinction between what is an uncontrollable stressor and what is a controllable stressor, or what is perceived as uncontrollable versus controllable. That can have a very large effect on how the stressor in the immediate term and in the long term affects whether it's a rodent or your patient. So I'm going to go into detail about these in the next slides. Another thing is previous experience with stressors that were perceived to be controllable. There's some very, very interesting research showing that in controlled studies where you have again, rodents, previous experience with a controllable stressor actually causes a positive, resilient phenotype. So the behavior of these animals that were previously exposed to stress, but it was a stress that they could sort of use adaptive behavior to get away from, that in and of itself has biological effects in the long term that create resilience. And another very important factor is, of course, developmental stage or age. There's a lot of evidence to indicate this is beating a dead horse or preaching to the choir, but I'm sure you all see in your work every day that obviously children and adolescents are much more susceptible than adults are to the long-term effects of stress. And then genetics, I just want to briefly touch on that. So we come back now to this diagram that I talked about earlier, which is representing the brain structures involved in the acute stress response. What I want to talk about is how does this diagram or this circuit actually change its behavior depending on the nature of the stressor. Sorry. It won't be immediately clear why this is relevant, but it'll come together as we go. So, as I said before, you have when you have a stressor in the environment, it leads to this activation of the serotonin system, which then causes more downstream negative effects, right? And I was saying how it's a very automated process. Well, what Mayer is, one of his famous contributions is sorry, I just wanted to add that the downstream hypothalamus effects as well. So one thing that Mayer determined is that the dorsal raphe nucleus, the serotonergic center, 5-HT is the abbreviation for serotonin, it naturally has this self-inhibitory behavior where the release of serotonin just in the local area around it actually causes inhibition of itself. So it sort of is an inbuilt way to make sure that the stress response doesn't just go completely out of whack. And what happens, this is something that Mayer determined, is what happens with chronic stress, uncontrollable stress, is that this repeated release of serotonin and then repeated inhibition of itself just at high levels compared to healthy organisms, what happens is it actually downregulates. So it desensitizes itself. So this is one of the ways that it's getting a lot into the weeds, I know, but it's just one of the very well defined ways that we know of that the stress response can predispose to further stress and further negative outcomes. So what happens is these particular serotonin receptors on this serotonin center, they're desensitized by repeated activation. And so what that does is it sets up this system for now any perturbation to it from stress is going to result in larger stress response. And we actually see this in rodents. If you stress them out repeatedly, like I described earlier, they end up displaying a greater stress response in their behavior as well. But now here's the hopeful message here, that another thing that Mayer has worked on is he discovered that the nature of the stressor drastically affects this this pattern of activity. So what he found is that we're not really sure how this works exactly, but it may be that prefrontal regions have the ability with other brain structures to recognize when a stressor is a controllable stressor or not. So in this case, they'll study rodents where the rodents are being shocked and they can actually figure out a way to turn the shock off. So that would be the picture to paint to actually understand how this is studied. But the evidence, molecular evidence as well as imaging evidence shows that the medial prefrontal cortex actually activates when there's a controllable stressor, or maybe more accurate to say is that it activates when the animal or a human realizes that a stressor is controllable. And this region has direct inhibitory synapses on the dorsal raphe. So this is arguably the primary way that a stress response is inhibited in favor of more adaptable behavior or in favor of simply health. And so, yeah, as I was saying, that this can be viewed as a key pathway for all treatment interventions. The dorsal raphe does not have a lot of inputs other than these limbic system inputs. really the only input from higher centers of the brain are from the ventromedial prefrontal cortex. Now, another thing that, of course, impacts the effects of stress, you all may be very familiar with this, but there is an interesting nuance to it that I want to point out. But first of all, I just want to say that if you look at brain development, what is always the case across species and in humans is that the lower centers of the brain develop first. The sensory motor cortex, for example, and the association cortices. So learning what is what, learning where things are in your environment, making associations between those. And then the last to develop is the prefrontal cortex. And during this time where the prefrontal cortex is having to integrate and actually regulate these other processes, there's also like during adolescence, that's, you know, the highest period of hormones, which all have effects on the neurobiology. And also during this time is one of the heaviest periods of myelination. So the neurons are myelinating their axons. In other words, the fibers that they use to connect and make meaning and regulate, those fibers are being formed and improved. But the important thing about this, also, again, the hippocampus here. So the hippocampus, neurogenesis is occurring at all throughout these periods. And like I said earlier, that's very important for using context to actually regulate responses in an adaptive way. And as I said, stress can have a directly negative impact on the hippocampus. But so what's interesting about this is that, actually, you know, I touched on this earlier. I'm just going to skip ahead. So that nuance that I was talking about is that pre-adolescent people, they actually do not display some of the stress responses or effects of stress on goal-directed behavior that adolescents do. So what this is showing is that if you have a laboratory model of learning fear associations, adolescents have the least ability to actually learn to, as I talked about with this pathway in the brain, they have the least ability to habituate their fear response via some sort of inhibitory messaging from the prefrontal cortex. And that is most likely related to this developmental just mixture of all these hormones and the brain trying to integrate prefrontal functions with the rest of the brain. Unfortunately, it makes adolescence, it makes for these negative effects. So another thing that we see in adolescents, but not in children as much, is increased difficulty with cognitive behavioral therapy. And that may be due to this, this soup of all of these developmental things happening at once and not having an efficient integration built in yet. Another thing I just wanted to touch on is genetics can also affect or moderate the effects of early life stress or even acute stress. So this is just showing that in humans, so BDNF stands for brain-derived neurotrophic factor, and it can come in different forms, but it's essentially a hormone that increases neurogenesis. Specifically, it helps with neurogenesis in one of the primary brain regions that has neurogenesis, which is the hippocampus. So the different polymorphisms or subtypes of this, the gene that codes for that hormone, can have different effects. So in those with the val-val version as opposed to the val-met version, they have a lower skin conductance response. They sweat less during a fear conditioning and fear extinction paradigm. And then they're able to extinguish that response or habituate via those adaptive signals of the ventromedial prefrontal cortex, more so than individuals with this val-met polymorphism. And the basis for that is that the val-met polymorphism is less active, and so that leads to less neurogenesis in the hippocampus. And there's some very distinct research showing that neurogenesis in the hippocampus is actually essential for learning to contextually regulate fear responses. So this is a very important hormone. And another difference that we see is that even in therapy with CBT for PTSD, individuals with these polymorphisms, you can take individuals who have the exact same amount of PTSD symptom severity and perform CBT, and those with the val-met polymorphism, they have a, sorry, I'm kind of confused here. These look like they're, anyway, follow up with me on that. But the takeaway is, I might have interpreted this one incorrectly, but the takeaway is that those with the lower activity polymorphism have a more poor response to CBT. And I won't go through all of this, but just for your entertainment, or if you want to do some follow-up reading, there are, even in this original study that was linking these adverse experiences to these negative outcomes, they attempted to link all the negative outcomes that were observed or associated with early life trauma. They attempted to link those two things biologically, and these are just some of the points that they considered that helped them, at least, this is 11 years ago, before we had a lot of the really good research in rodents showing the just how strong these effects were causally. These are just some of the points that they wanted to highlight that made them think that this is indeed a causal relationship as opposed to a correlation. And now I want to turn to talking about how do treatments actually engage the adaptive systems that I've highlighted. So this is just reiterating that in fear extinction paradigms, which we use to study exposure therapy, ventromedial prefrontal activation is predicting success and actually inhibiting that fear response. And then this comes out of our lab with Dr. Sisler. Josh Sisler was my former mentor. He's in Wisconsin now. But a lot of you probably know him or of him. But he, what he found is that he did an fMRI-linked therapy study where he had, I think it was 37 adolescent girls who had a history of trauma. And over the course of a year or so, he gave them repeated exposure therapy sessions, or they had exposure therapy with other clinicians. And along with that, he did fMRI experiments. So he showed that those those adolescents who had the best treatment response were actually those who, it's not that they had, the important thing is not that someone has the least fearful response or the least activation of the amygdala during some sort of fear stimulus, but it's actually how well they can discriminate between a fearful stimulus and a safe stimulus. So again, going back to the hippocampus and actually properly contextualizing responses, it's those who could properly contextualize or who had the most different responses to fear versus neutral stimuli that had the best treatment response. So in this case, you can see that I believe the red is the those who didn't have a great treatment response. You can see that they kind of have this positive amygdala activity in response to all of the stimuli, whereas those who had a good response to therapy, and this is controlling for initial symptom severity, those who had a good response to therapy didn't have much of a response to the neutral stimulus. They had a much greater response to the fear stimulus. And then, as you can see, they're actually discriminating fear from safety very well. So that goes along with that theme of being able to contextually regulate. Now, these are just kind of a shotgun of some very interesting links between all the biology that I outlined and the treatments that we know are effective, but we're not exactly sure how they work, just because the brain is so complex, obviously. So these are just some of the clues. So for example, we know that stress inhibits hippocampal function. There's a specific function of the hippocampus, which is to form synapses and actually regulate prefrontal activity, which is that formation of synapses through learning is called long-term potentiation. SSRI treatment restores long-term potentiation in the hippocampus. Again, it's not a complete picture, but it's pretty interesting nonetheless. And that should not be too surprising because SSRI treatment also increases that brain-derived neurotrophic factor, which I highlighted earlier, as important to the hippocampus forming new neurons and synapses. And kind of working backward in causation here, the hippocampus also increases its neurogenesis and volume with SSRI treatment. And someone has actually shown that the level of neurogenesis predicts the level of improvement in depression, for example. SSRI, chronically, they also reduce amygdala activation of fearful stimuli. There is a little bit of conflicting data regarding how SSRIs interact with fear behaviorally and in the brain, but I just wanted to highlight this one point. Another thing that SSRIs do is they, so as I mentioned many other times in the talk, serotonin, surprisingly, is released during acute stress and causes a lot of these downstream negative effects. One of the ways that it does that, as I've said, is through the hypothalamus, but those receptors on the hypothalamus are a specific type of serotonin receptor that if you acutely stimulate this type of receptor, it causes anxiety, and it also is responsible for that hypothalamus pituitary axis stress response. One thing that SSRIs do is they desensitize that that stress response through that system. And
Anthony?
it also increased synthesis of a brain neurosteroid called allopregnanolone. You don't need to know that name specifically, obviously, but that's interesting because this brain steroid is responsible for potentiating the effects of inhibitory neurotransmission. So for example, Xanax and Valium and other benzodiazepines, they work by increasing inhibitory signaling through GABA. And allopregnanolone actually enhances that effect. But anyway, I don't want to act like pharmacological treatments are just the end-all be-all.
Anthony?
Yeah.
Hey, I'm going to have to forward it to our CU. I know we're right at the top of the hour, and for folks that have to leave, so they can get the CU information, and we can hang around for a couple minutes to answer any follow-up questions. Is that okay?
Yeah.
Okay. Kind of get, there we go. Okay, so for folks who are interested in getting the CU, the code's displayed there. Be sure to email me, and if you tell me your first and last name, that's just really helpful. It makes it easier for me to find you in our registration list. If you're watching as a group, just let me know who the individual was that signed into the webinar, so I can, because obviously if you're watching with a partner but you didn't sign in, I won't see a history of you on our attendance list. So I'm going to have this slide up here for a little bit, and if you have any questions, feel free to kind of type those in. Anthony, thank you for such a really interesting presentation. I learned a lot.
Great.
You know, it's always interesting to kind of hear the ACE study, and then how that all it's really cool to see how the biology is kind of catching up with that. What was
Yeah. So. I hope it was helpful. I hope I didn't go too into the weeds with the neurobiology. If everyone can still hear me, the one thing I wanted to clarify there at the end is that cognitive behavioral or exposure-based cognitive behavioral therapies are definitely the most effective, according to the statistics, treatments. And
can you talk a little bit more about that? Like, what are their specific names? Do they have a specific name? Because many of your folks have trained in trauma-focused CBT, but what are some other evidence-based treatments that are exposure-based that you're referring to?
So there's, I've got a list. Let's see, actually I can pull it up here. 1 second. So there's prolonged exposure therapy, there's simulator-based exposure therapy, there's sorry, you can tell I'm not a clinician, but those are the only two I can come up with offhand, but the literature is clear that, I mean, maybe if you guys want to look up other types of exposure therapy, maybe that would help, but the literature is clear that exposure-based therapies, sorry I can't name examples, but they are the most effective, even compared to other types of cognitive behavioral therapy.
Okay, thank you. And I know we do have a couple of questions that have come in here, but in the interest of time, Anthony, I'll probably forward those to you, and then we can follow up with those participants following the webinar. So,
yeah.
And I'll just, hopefully if most folks have kind of got the CU information, I'm going to back up as one more kind of final plug for Anthony's study. I know many of you do work with children, but children also have caregivers and adults in their lives, so perhaps maybe mom or caregiver between the ages of 21 and 50 has a history of assaultive violence. Be sure to, you can email me if you don't forget to write down Anthony's contact information there, and we'd be happy to share more information and what's all involved with that.
Yeah, thanks a lot, Jed.
So.
And
so I'm going to, yep, go ahead.
Yeah, just one quick note, if people are interested, this is actually looking at how dopamine can reinforce the consolidation of safety memories in exposure therapy. So it's a very novel and well-supported hypothesis. So I hope we can get some more participants because we're getting some really good data. But thanks for that, Chad.
No problem. I'm going to sign off here, but I'll leave this slide up for a couple more seconds and advance it to the CU. And thank you everyone for joining us over your lunch hour today. Take care.
Thank you everyone.